school's out, blame corona, but I still got some science I could show ya. No class, no school, that's cool, throw the class on the YouTube. Even if they shut the school down, I'll get that knowledge anyway somehow. Real scholars won't stop for corona. Tune in for that science takeover.
Happy Wednesday. Welcome to Mrs. Finney's science class. Shout out to our HB Warriors, to all of our subscribers, and to all of our visitors. From my home to your home, let's learn science. Today, after our wrap up from yesterday for lesson 8, we now want to read a little further into this information. So let's take a look here. The reading further section is titled Surprises in Our Genes. Learning objective for today, I can explain the Human Genome Project. Have you ever heard of this project before? What in the world is it? We'll find out today. So let's take a quick look at this section. It says in 1990, sounds like a long time ago, right? In 1990, one of the most ambitious scientific efforts in history was launched, the Human Genome Project. What was its goal? It aimed to locate the positions of all the genes that instruct for making proteins in each of the 23 pairs of chromosomes in the human body, a process known as mapping. Scientists knew the undertaking would be enormous and take many years. The surprise was that much of what they thought they knew about genes was wrong. Go figure. The gigantic Genome Project. By 1990, scientists knew about genes and where some genes were located in the chromosomes of a few species. But now they wanted a complete map of the human genome, all the genes in a person's body. This project, called the Human Genome Project, HGP for short, was supposed to take 15 years. That's a long time. But the HGP's goals were very ambitious. If you think of a gene as a word, it would be made up of a series of letters. To read the gene, you have to know what those letters are and in what sequence. This process is known as sequencing. If you think of the chromosomes as a sentence, knowing which words or genes come in what sequence is important for understanding the sentence. This is called mapping. Human chromosomes are made of 3 billion individual letters of DNA. Sequencing those letters and mapping the genes was an enormous task. The U.S. government funded the program, and institutions in 5 other countries all worked alongside the United States to perform the work. At the same time, a private company worked hard to race ahead of the government-funded groups. Scientists often cooperate in their research, but sometimes they compete as well. Whoever mapped and sequenced the human genome first was sure to go down in history, and many different people wanted this honor. Because each gene can have several variations called alleles, the DNA sequence among individuals varies slightly. DNA samples from people of different ethnic backgrounds were collected, broken up into smaller DNA segments, and stored in DNA libraries that will become available to scientists for mapping and sequencing. Finishing the project thanks to technology. Then the enormous task of sequencing the DNA began. How could they possibly gather all that data? The job was too big to tackle by hand, so scientists relied heavily on technology to help them collect and analyze data. But computers in the early 1990s were nothing like they are today. They couldn't handle huge amounts of data, and they were extremely slow. So an important part of the HGP effort was developing and improving technologies that could assist in the research. These included faster computers that could process huge amounts of data quickly, robots that could manipulate DNA samples, and an internet more capable of data sharing. In this way, the research drove the improvement of technology, and the technology in turn drove the research. Science and technology often work in this way, with one spurring on the other. Once the technologies improved, the work went more quickly than anyone expected. The first completed chromosome map for chromosome 22 was announced in December of 1999. By May 2000, chromosome 21 had been mapped. Only 1 month later, a rough draft of all 23 chromosomes, the entire human genome, was completed. Although the Human Genome Project was expected to take 15 years, technological advances resulted in publication of a final, high-quality map of the genome in April of 2003. 2 years early and under budget. In the end, both the private company and the government decided to work together and release the human genome for the whole world to use free of charge. Check that out. Before the HGP began, scientists thought they knew what the human genome map would basically look like. They expected to find that human genome had many more genes than the genomes of other animals, since humans are so much larger and more complex than many other species. But wow, were they wrong? Unexpected results. Some of what scientists already knew about human genes was confirmed by looking at the completed genome map. But there were a few big surprises. Number of genes. One of the biggest surprises was that the human genome consists of only about 20,000 genes that are in the instructions for proteins, not the 100,000 or more that scientists had expected they would find. How does the size of the human genome compare with those of other species? A fruit fly has about 13,000 genes. A roundworm has about 20,000. And mustard weed has about 27,000. Our genome is worm-sized? Genome size doesn't seem to be related at all to an organism's size or complexity. Go figure. Genes and traits. Scientists had also believed that each gene was the instructions for only a single protein. What they discovered, though, is that most genes are the instructions for more than one protein. If a cell's gene reading machinery tells the cell to read the instructions a certain way, one version of the protein is made. If the instructions are read another way, a different version of the protein is made. This means that a single gene can code for more than one trait. The HGP also verified previous data that a trait can also be determined by more than one gene. Junk DNA. Another surprise was that not all DNA codes for traits. In fact, large segments of DNA seem to be junk. That is, they don't contain any genetic instructions, but this junk DNA may not be junk after all. Scientists now think that some of these segments act like on and off switches to control genes. When a gene is turned on, it makes a protein. When it is turned off, no protein is made. Similarities among humans. Researchers were surprised at how similar humans are to each other, considering how different they look. Only 0.1% of the genome makes two unrelated people different from one another. Go figure. The genetic difference between two alleles of a trait, such as eye color or the presence of freckles, is often a difference in only one DNA letter. Wow. Similarities to other species. Scientists used to think that humans were very different from other species, but it turns out that we aren't. Actually, humans share 98% of their DNA with chimpanzees, 65% with chicken, and 26% with yeast. Go figure. Here's a visual. Percent of DNA in common with humans. Check this out. All right, so we have humans, we have different humans. 99% similarity, chimpanzee 98, mouse 85, dog 84, zebrafish 73, chicken 65, fruit fly 60, roundworm 35, baker's yeast 26%. Wow. So what does it mean to have 98% of the same DNA as chimps? Humans certainly aren't as hairy as they are. However, our basic body plans are the same. 4 limbs, 2 eyes, a nose, 2 ears, and a mouth. Our basic life processes also are the same. We digest food and reproduce the same ways. But what do humans have in common with unicellular yeast? The cells of both species have similar cell membranes, cytoplasm, and organelles. Both species take in oxygen and use it to process food and release energy. We are more like a simple yeast cell than we might imagine. Scientists were wrong about what they expected to find in a human genome, but that's what science is all about. It's okay to be wrong. A society has benefited from what scientists have learned. Better tests for identifying genetic disorders have been developed. Cancer treatments are being customized for individuals according to the differences in their genome that affect how the disease responds to certain medicines. Although much of the genome secrets are still unknown, the Human Genome Project has been a huge success. I hope you enjoyed this reading. Oh, we're so close. All right. Thank you for tuning in. Just want to show you this last piece here. Look at this. See? We're more alike than we might think. More alike than we might think. Go figure, right? Variations of traits make humans look very different from one another, but their DNA is actually very similar. Individuals have more than 99% of their genes and alleles in common. Wow. After all, we're more alike than we may have even known. How beautiful is that? We're so close. Even when we're far away, we're so close. All right. Thank you for tuning in today. We want to wrap up with our outro. Here we go. It's been fun. It's been real. We break it down and pick up science skills. Give yourself 2 claps just for tuning in. Come back, meet tomorrow. We can learn again. Oh, it's been fun. It's been real. We break it down and pick up science skills. Give yourself 2 claps just for tuning in. Come back, meet tomorrow. We can learn again. Thank you for tuning in. Have an awesome rest of your day. Peace.